I really wish I was in Sydney right now and not sitting at home in my loft, but in any case, thanks very much for turning up. So before I start my talk, I just want to thank my co-authors. It was a really interdisciplinary effort, this piece of work. Corinne is a quantitative ecologist who specializes on Weddell seals. Jenny is a statistician and Lars is an oceanographer, and I'm sort of a bit of everything, I guess. Right. See how I can move my slides along. Oh, there we go. Great. Okay, so I'm just going to start by talking a little bit about Weddell seals and then their namesake Antarctic shelf sea, as well as the research questions that we set out to answer, how we did it, and some conclusions. I'm not going to talk about hidden Markov model details at all in this talk. There just isn't enough time. Plus, there's a brilliant skills showcase that went on yesterday evening, which I didn't see because I was asleep, but I know for a fact VNA will have done a brilliant job. So if you're interested in knowing more about hidden Markov models, then please go back and look at that. Okay, so this map that you can see here is a map of Antarctica. The Weddell Sea is quite a big area. It actually covers this whole area here, but in the yellow box is where my tracking data came from and that I analyzed for this study. And the data came from Weddell seals that are pictured here using these animal-borne satellite link devices that record behavior of the animal as well as salinity and temperature of the water that they're swimming through. Okay, so Weddell seals are large foetid seals. They stay in the Antarctic pack ice all year round, and they use the ice to rest on a daily basis, and they dive deeply down to 400 meters or more, and they hold underwater territories in the breeding season. So they really utilize this very diverse, highly seasonal, highly dynamic environment of the sea ice, which can be quite open looking like this at the end of summer, or it can be completely closed. So the Weddell Sea, and why did we study Weddell seals in the Weddell Sea? Well, it's quite different from most of the other Antarctic shelf seas in that it's got this wide continental shelf and it features an extensive winter sea ice. And together these two things interact to contribute to the formation of Antarctic bottom water here. And I'm just going to spend a second talking about that. So this is a picture of the Weddell Sea. So this is what is land and ice in the yellow, and then the paler green is the shelf areas, and the deep blue is the deep ocean. So what happens here is that you get sea ice formation on the shelf, you get these southerly winds that push it offshore, and that essentially exports the freshwater component that's bound up in the ice, and what's left is the salty component that mixes and contributes to higher density waters that sink to the bottom. So you get this high density water that's sinking to the bottom in this area. So what you end up with is this seasonally varying, highly stratified, vertically stratified water column with a variety of available water masses that carry different kinds of prey, etc. So I'm going to show you a picture of the kinds of water masses that you get in the Weddell Sea. This is what oceanographers call a TS diagram, or a temperature on the y-axis, salinity on the x-axis diagram, and these contour lines are density. So here you get these very salty, very cold water masses, and they sit on the shelf because they're really heavy, they sink to the bottom. So I'm showing you this because later on I'm going to come back to it and show you not what a ship collected in the Weddell Sea, but what the seals collected. So of these available water masses, what did the seals actually visit? So the data we had came from 2011. We had 19 Weddell seals that were tagged in late February and collected data into June. There were 10 females and 9 males, so this was a nice split that enabled us to look at sex-based differences. So in this map, you can see the female tracks in the sort of pinky salmon color and the males in the more purpley color. And you can already see that the females range quite widely off the shelf, whereas the males stay pretty much exclusively on the shelf. So coming to our research questions, we wanted to know what kinds of dive seals carried out in this area. We wanted to know if certain dive types are linked to water mass. And we also wanted to know if there was a seasonal or a diurnal pattern. And this is all based on information that we know from Weddell seals in other areas, and also sort of specific questions to this area. For example, we've got a variety of water masses. Are seals using those water masses differently in the Weddell Sea? So we answered this question with HMMs. It's not the only way, but as far as I'm concerned, it's the funnest way. Okay, so if you haven't seen a dive profiles of seals before, this is what it looks like, where you've got depth on the y-axis. So these flat bits here are surface periods, and you get tiny little dives, and you also get these deeper dives, like these are going down to about 350 meters here. So we extracted some metrics from these from this sort of time series to use as state-dependent variables in the HMM. So I'm going to start with behavior duration. So how long did a behavior last? And I say behavior, not dive duration, because we also included non-diving behaviors in the time series, so haul outs and surface periods, and duration was the only thing that was characterizing those because they're not dives. So for the dives, we extracted hunting depth using this method developed last year by Corinne here and her colleagues. We looked at proportion of time in a dive spent hunting, the proportion of bathymetry reached during bathymetry reached at hunting depth, so the hunting depth divided by the bathymetry, and the salinity at hunting depth. And we used all of them here. And I know it's not common to use a box, but I just couldn't get them all to fit in a circle. So these are the state-dependent variables, and they're informing the underlying state. So the movement states we were interested in, we knew that haul-outs and surface events occurred, but we weren't interested in their parameters per se. So these get provided to the model as known states. And then what we are interested in is finding out about the different dive states. And the covariates that we used were a local time of day and week of the year. And these covariates act on the probability of transitioning between states. So what kinds of dives do Weddell seals carry out? This is a sketch I made in my workbook a few months ago, just to try and get my head around what was actually going on. I've used it a few times. It turns out it's quite handy. So here on the left, if you don't know much about seals, this is a quick what seals do. So seals at least Weddell seals spend a lot of time hanging out on ice, doing not much. We call this being hauled out. This is a non-diving state. And they also spend quite a lot of time at the surface where they're not diving, but they are in the water. And this is basically up until the dive trigger threshold. So below 6 meters, they're considered to be diving. So this at surface state is when they're at the surface, they're in the water, but they're not diving. We weren't interested in knowing about the parameters of these states, so we provided them as known. And we found that, because we were interested in these sex differences in this rich data set, we found that actually both sexes do these 3 types of dives. One of them is really short, shallow. This is not drawn to scale, I'm sorry. This is 12 meters. So really short, shallow dives. They also do these dives down sort of within the first 100 meters of the water column. I call these epipelagic dives. And then both males and females do these deep dives that actually go really close to the bottom. So these are deep benthic dives. However, we found, oh, here we go. So here are the dive types in common. However, we found that in females, there's this extra state, which we don't find in males. And it's sort of an intermediate state. It goes down to about 200 meters and it doesn't reach the bottom. It's very far from the bottom. So this is actually a pelagic dive type. So that we've got this unique female dive type that are pelagic dives. So now we know what kind of dives seals do. So are these linked to water mass? And you'll have noticed before that salinity isn't a covariate because these environmental conditions aren't something that the seal's going down and sort of using as a cue every time it dives. These are long-lived animals. They've got complex spatial memory. It's really unlikely that they're using these abiotic conditions to sort of decide on a dive-by-dive basis what they're doing. For me, it's actually more part of what the behavior really is. So, and you can see that these behaviors segregate in temperature salinity space. So just casting your mind back to the figure from the ship-based surveys where you've got all the different available water masses. Here we've got what the seals chose. So in the pale green and darker green, you've got the quite shallow surface states, so down to 100 meters. And this is what you would expect. There's a narrow range of temperature and salinity that you can get at the surface. So this is what we would expect to see. However, once you get to the deeper dive types, pelagic and benthic, you see that they're segregating in depth. So you can see here, both the males and the females are using these high density, high salinity water masses that you find on the shelf in these benthic dives. And then in the females, you see that the pelagic dives are actually using a totally different water mass. This is what we call modified warm deep water, and it's a mix of warmer water masses that come in from the north into the bay embayment that is the Weddell Sea and mix with these cold water masses on the shelf. And these occur away from the coast. This water mass occurs away from the coast. And as you can see, this is pretty much completely absent in the males. And that sort of further justifies why we weren't able to fit a 4th dive state in the males. The model was just numerically unstable. So here we can see that the different dive types are actually separating in temperature salinity space. Essentially, these are different types of habitat. So lastly, is there a seasonal pattern? So I'm hoping I can put across what this plot says. Basically, we worked out the probability of finding a seal in a state according to some conditions. On the x-axis, we've got local time of day, and on the y-axis, we've got week of the year. And this is week 7 was the first week we tagged the seals, and week 24 is basically the solstice. In fact, it's this week we're in now. So, and week 15 of the year is about April. So we find that in late summer in the Antarctic, female seals are doing these pelagic dives pretty much throughout the day with a bit of a peak in the afternoon, and benthic dives are centered around local midday. As the season progresses into week 15 and week 24, we see that pelagic dives actually become more peaked around midday, the same as benthic dives. And then towards week 24, both of these deep dive types kind of reduce in likelihood, and what you see is these epipelagic dives are much more likely. If we look at the males, obviously they don't have this pelagic dive state, but you see that this is actually the confidence intervals around the probability of being in a benthic dive are narrower for the males. And this whole picture is basically that the males are much more aseasonal, which is interesting in itself in terms of what are the ecosystems that these two different, these sexes are exploiting, these different groupings, interspecific groupings. So just to finish off, the conclusions: females and male Weddell seals share a number of dive types. They both dive into cold bottom water using benthic dives. Females only also dive into the warmer water masses away from the coasts using pelagic dives. And there's this clear seasonally varying diurnal pattern in behavior. And we find that the males stay on the shelf pretty much the whole period that we examined, from late summer into winter, whereas females leave the shelf and dive pelagically. And if you're interested in knowing more, this is up as a preprint on bioRxiv, and thanks very much for listening.